Welcome to video 2 in the female reproductive endocrinology lesson. This video will start the process of talking about gametogenesis, that is development of the gamete, that is the ovum, and folliculogenesis, the development of the follicle in the ovary. These two, as you will find out, go together. And as the ovum develops, the follicle develops in parallel through stages. So this is the process of gametogenesis in the female in a nutshell. We start with the primordial germ cells and the development of oogonia. And the start of this process begins with a round of mitosis, both in the male and in the female. You can see them in parallel here. We go through, after that, 2 rounds of meiosis, 1 and 2. And eventually, the process finishes with the production of your gametes. You have your spermatozoa on the male side. You actually have your ovum here, which is arrested until fertilization takes place. And at fertilization, the last meiotic phase completes, and you have a fertilized ovum at the end of this. And during this process, we go from a diploid germ cell oogonia to haploid gametes. This process, of course, ceases at menopause in the female. It is different from the male in this case, in that the process doesn't technically cease completely in the male. Again, we have this phase of mitosis going on. It certainly takes place in the embryo, and we get initiation of meiosis. And this first meiotic division is arrested at the diplotene stage, pre-puberty. So before puberty in the female, we have a lot of oogonia that are arrested right at this stage, right where they're ready to continue with meiosis and the generation of the ovum. This is the primary oocyte, and you will see that we have really what comes down to 2 full sets of chromosomes here, and we have stopped during that first meiotic phase. After puberty, meiosis resumes, and the first meiotic division takes place, and now we're down to 2 chromosomes here, and the other 2 here are contained within this first polar body. So we have half the genetic material getting all of the cytosol, and then this polar body is basically going to be genetic material that we are going to throw away. We enter a second meiotic division, and as I said, that doesn't complete until fertilization, when we get a second polar body that forms here. So half of this genetic material is now going to end up here. We have 1 chromosome, and it is going to pair with a set of chromosomes from the sperm. And so we have genetic recombination and fertilized ovum. Okay? So for the male, we don't have this arrest until puberty. It just goes straight through the line pretty much all the time without stop. So for the numbers game, we have 7 million oogonia by 20 to 24 weeks of fetal development. So we have a whole bunch being produced in the fetus. At birth, this is down to 2 million oogonia, and this is due to and then we're down to hundreds of thousands of oocytes at the start of puberty, and this decrease in the number is due to a process called atresia, which is basically when these oogonia die off in the absence of gonadotropins. So without gonadotropins, these oocytes can't develop further, and so they just regress and die off. They basically undergo apoptosis until you hit puberty. And so you have a decrease in the number of oocytes that are available after puberty to ovulate and potentially develop. Okay? So that being gametogenesis, we can move on to the related process of folliculogenesis. This is, quote, an ovary, unquote, but please don't make the mistake of thinking that this is anatomically and histologically correct. It is not. It is a cartoon. In this corpus luteum, for instance, substantially larger relative to the size of the ovary than a real corpus luteum would be, that is for sure. Everything before stage 2, so the development from a primordial follicle through the primary follicle to a secondary follicle, that takes place before puberty. So at puberty, we have a bunch of follicles that have developed along with hundreds of thousands of ova inside them that are frozen right here at the secondary follicle level. Post-puberty, when gonadotropin levels begin to become significant, you catalyze stage 3 of the development of the mature follicle, and 4, which is ovulation. You get development of course, corpus luteum, which we'll talk about considerably later in the lesson. We'll concentrate on that at the moment. And then you end up with an atritic follicle, which is basically what's called a corpus albicans, which is just scar tissue, more or less, on the in the ovary. Okay, and this is assuming, of course, that pregnancy does not take place. So, if we take a little bit of a closer look at this development, we have our primordial follicle up here. This is sort of the development of the follicles from that primordial stage to the Graafian follicle, which is what we call a mature follicle. We have notice right away these granulosa cells grow and proliferate along with thecal cells, and these are your primary generators of hormones. And so as this follicle grows, you're producing more and more hormones, which are involved intimately in the menstrual cycle. As we progress to the primary follicular stage, we have the development of the zona pellucida, which is developed from granulosa cells here. They secrete a mucus layer sort of inside and around the inside of the follicular layer, and this forms a barrier between the granulosa cells here and the ovum here. We also have the development of a theca interna, and the follicular cells are producing estrogen. The theca interna are going to be responsible for producing androgens, and once again you need these androgens in order to develop estrogen. So these two layers of cells work together quite closely to produce estrogen. You also have some granulosa cell perfusions, which are extending to the ovum and providing nutrients to the ovum. We then progress to the secondary follicular level, and here we see the development of antrums here. And these are formed by exocytosis of droplets, liquid droplets from the granulosa cells, and that liquid is actually fairly important. It has a lot of hormones in it, including FSH and LH, estrogen, progesterone, perhaps most importantly, oocyte maturation inhibitor. That inhibits meiosis, and of course at the secondary follicle level you stop developing, and this is, of course, where you end up at the pre-puberty stage, where you're part of the way through that first meiotic division and you've stopped, and this is largely due to the existence of this oocyte maturation inhibitor. Okay? And then we also have the development of a hard theca externa. This is a fibrous shell that protects the follicle. And then finally, after puberty, you develop into a mature Graafian follicle. You have the follicular cells, which develop this membranous membrana granulosa. The antrums here coalesce and form what's called the liquor of folliculi, and that surrounds the corona radiata, which are follicular cells that surround the ovum, and then that's connected to the membrane granulosa by this cumulus oophorus here, which sort of holds the ovum in place in preparation for ovulation, when this whole thing's going to break apart and this ovum's going to be free of the follicle. So if we look at these stages one by one, we have the start at initiation, and we progress to the secondary follicle, and this is where the pre-puberty stage stops, at stage 4. Okay, so stages 1 through 4 don't require gonadotropins, which is why they take place before puberty. They do happen throughout the life of the female, not just pre-puberty. You'll notice, but none of the stages that happen post-puberty take place before puberty, obviously. So everything goes to this stage and stops. Notice the substantial increase in size here. We start at 0.03 millimeters. We end at about 2 millimeters. That all takes about 200 days to take place. Okay. Stages 5 to 8 happen under the influence of the post-puberty hormonal changes, including the existence and of the gonadotropins. So many follicles are recruited at stage 5. So you get quite a number of follicles recruited here, but eventually these are selected, and eventually selection takes place, and one dominant follicle ends up out-surviving the rest. Okay, so although many follicles start at stage 5 at once, you end up with one follicle that sort of rules them all, and the rest of the follicles sort of regress and die off. So this is our post-puberty to menopause. These stages take place constantly after puberty all the way until menopause starts. And you'll note the proliferation of the granulosa that takes place here. We're talking about, GC is granulosa cells. We're talking about 10 to the 30 here. All of a sudden, we get into these stages where we're ending up with 10 to the 6th. So, 1,000 times more granulosa cells, and that means a lot more estrogen can be generated here as we progress through 47 times 10 to the minus 6th granulosa cells. Okay. So what does that mean? Well, it has a lot to do with the process of selection and how one dominant follicle emerges from the rest. So first of all, let's talk about some of the other functions of the estrogen. It alters cervical mucus to enhance sperm transport, so it becomes more liquid and less acidic. Peristalsis begins in the fallopian tubes. Again, you bring the sperm to help. That sort of helps bring the sperm and the ovum together to help fertilization take place. And you have movement of the fimbriae here, which is stimulated by estrogen. And the endometrial layer proliferates under the influence of estrogen. Sort of prepares the uterine endometrium for progesterone, which is really going to be where a lot of this proliferation is stabilized. takes place. Estrogen, as far as selection goes, we get inhibition of growth of cohort follicles as the dominant follicle is selected, and these die away. And what seems to be the most important determinant of what becomes the dominant follicle and what becomes all the rest, which are sort of inhibited and die away, has to do with the FSH receptors, and the dominant follicle appears to be the one that has the most FSH receptors. So what's happening here is you're secreting more and more estrogen. And as you guys will recall, estrogen feeds back upon the hypothalamus and upon the pituitary to decrease gonadotropin secretion, FSH. So as more and more estrogen is produced by these follicles, the ability to respond to FSH becomes more and more important. So the more FSH receptors you have, the greater ability you have to respond at low levels of FSH. So the dominant follicle appears to be the one with the most FSH receptors that can respond to the lowest level of remaining FSH in the presence of estrogen-mediated negative feedback. And so more FSH receptors seems to be the determining factor for which follicle becomes dominant, and which of the cohort and which one survives as the cohort follicles are inhibited and regress away. The estrogen also primes the hypothalamus to secrete GnRH, and it increases LH stores in the anterior pituitary. This is going to become very important when we go into the process of ovulation, because this is initiated by what's known as an LH surge. And that LH surge needs a lot of LH stored away in the anterior pituitary, and it's going to need a lot of GnRH generated to stimulate the release of that LH. So these two are necessary for what we call the LH surge, and that's going to happen when the negative feedback of estrogen upon the hypothalamus and the anterior pituitary becomes positive, and you get this huge surge of LH. And in order to do that, you have to be ready to secrete a lot of GnRH. You have to be ready to secrete a lot of LH. Estrogen seems to have a role in preparing the hypothalamus and the anterior pituitary for that, and we'll talk more about that in a minute, and a little considerably more later. So that is the end of this video. We will continue on with this process in the next video, where we talk about it from beginning to end and how the process of ovulation takes place as a result of this development.